Thank you everyone for joining us back again, which is morning, slightly just before lunch here in Waterloo, and I know late evening as well as late night in other parts of the world as well. So it is my distinct pleasure to invite Professor Albert Vandenberg, who is the co-scientific director of MESA Plus, as well as the chair of NanoNext NL, to give a presentation from lab on chip to organ on chip for the session 4, which is related to the United Nations Sustainable Development Goals number 3, 9, and 11. Thank you so much, Albert, for joining us.
Okay, thank you very much for the kind introduction. Good morning and good afternoon and good evening to all of you who have been able to follow it. I have to apologize. Yesterday I was really very feverish because of COVID and I could not make it, but today I will try my best to give you a presentation about my work or our work at the University of Twente. This is an aerial view of the university with the MESA Plus Nanolab that you just heard existed this year. The Nanolab 10 years, MESA Plus 20, and MESA, that was the predecessor of MESA Plus 30 years. So we really have a long-standing history in the field of nanotechnology. I want to talk about something which uses microfluidics as enabling technologies, and the comparison with microelectronics is clear. I think microelectronics enables you to make a radio on the size of a chip, except for the loudspeaker. Microfluidics hopefully enables you to scale down a lab to the size of a chip. And in the past years, 10, 20 years, many technologies have been developed. These are different microfluidic technologies that have been made. You can see them all, and so the toolbox is ready. It's time to get to work and to do something with it. And that was also our vision in the past years. Let's see what we can do with it. And the first thing is already all the work that we did to develop a point-of-care system to measure lithium. Lithium is used by manic depressive patients, and we, together with a couple of PhD students and postdocs, we developed a chip that you can use with capillary electrophoresis to detect lithium in whole blood. And the is that you separate the ions by capillary electrophoresis. Here you see if you use a mixture of 3 ions, potassium, sodium, and lithium, you can separate it quite quickly. If you go to higher concentrations, more blood-like concentrations with high sodium concentrations, then you have to separate a little longer to get the lithium peak out of your shoulder of sodium, a very big peak of sodium. Lithium is used as antidepressant, and the patients want to measure it in order to make sure they have the right medication concentration. Now, this chip is quite big, as you see, 2.5 by 5 centimeters. By far not good enough, of course, for a point-of-care system, but together with the company Micronics and also a spin-off company, Medimate, we've been able to develop a much smaller chip, which is put in a plastic holder and could be produced. By the way, also the production was done by Micronics company. And here in the upcoming animation, you will see how the patient uses this point-of-care chip to measure lithium in blood. So the patient has, just like a diabetes patient, a finger prick to get a little bit of blood on the finger, put it on the chip, which is in the holder here, close the holder and put it in the instruments. And now you will follow what is happening here. The chip consists of 2 glass plates, a bottom glass plate, which basically has the white microfluidic channel etched in it. Top plate has the gold connections, and if you inject the tiny drop of blood, and in fact you only use a millionth of the drop of blood, and you put the electrical voltage, you separate the ions by capillary electrophoresis, and when you know that when the lithium ions pass between the electrodes, you can calibrate that, and you can measure the concentration of lithium ions quite easily. After a few minutes, you have your measurements, and then you know, as a patient, whether or not you are on the right scheme. So this worked well, and actually I also tried it on myself. Under tight control of a psychiatrist, I took a little lithium pill, and I could measure 20 times over time and see the double exponential curve rising and decaying of concentration of lithium, so that gives a nice indication. The good thing of this measurement is that it measures not only lithium, you get all the ions for free, you get all the other positive ions if you measure it, if they are present, and if you reverse the potential, you can also measure negative ions like nitrates. But in addition, you can also try out for creatinine, which is a good indication of early kidney disease. And also drugs like GHB can be detected using this technique. So it's a multi-use platform. So that is, let's say, an example of an already old example of a lab-on-chip system. I would now like to change gears, not to cells, that will be my last part, but I will move to organs on chip. What are organs on chip? Organs on chip, of course, are not complete organs, but they are miniaturized organs in biocompatible microfluidic chambers. And what they do is they mimic the organ physiology, healthy, but also disease conditions, and that's very important. And of course, what you also want is to combine more than one organ or organ on chip together, because they influence each other. If you have a kidney and a heart, or a kidney and a lung, or a liver and a lung, they can all influence each other, and you may also be interested in how these different organs on chip influence each other. The question, why do we need it, is answered here, because typically if you want to develop a new therapy, you need drug, you have normal testing on cells, or very complex testing on animals. The cells are very often too simplistic, and the animals are too complex and also not representative, because many animals do not have the human disease present in itself. So there is a need for something in between, and that's the organ on chip. You have a human model, you have a dynamically highly controllable microenvironment. You can do dynamic drug dosing, for instance. You can use it for drug screening, you can use it for precision medicine, but you also reduce the animal models. Nobody wants animal models, but they are the only ones that are available at this moment, and you cannot go around it. So here you see a typical view of the drug development funnel of the pharmaceutical industry, starting with 10,000 compounds. And you see that after the first phase of 5 years, there's a phase of animal testing, and finally the very expensive, where you see the dollar signs over here. Let me see here. That's the phase where you have the clinical trials. So these clinical trials cost you in the order of billions. And if you can limit the number of candidates in this second phase, where you do the animal testing, by using more predictive organ on chip models, you have less candidates left over in very expensive clinical trial phase. And that will save a lot of money and speed up the process of drug development. Now, what is basically the driving force also behind this is that we go from a before status where you have a reactive sort of healthcare system, where you first have symptoms, and then you have the symptoms based giving you diagnostics. You have non-translatable models, and you have non-specific treatments, to a new era where you have better understanding. You can have personalized models, and you can have precision medicine. So that is a proactive system where you can really predict better what is needed for a patient instead of reacting to things that happen. Now, over the past years, and in many institutes, I guess also at WIN, but also at the University of Twente, many organs have been made on chip. And you see a list here of the left-hand side of all the organs that are being tried out at our university only. And you can imagine that all these different organs require different microfabrication, microsystems, and it's not easy always to connect them or to make use of common technologies. So this is why we have developed a concept which is called the translational organ on chip platform. Basically, it's similar to let's say, the breadboard of electronics, where you have a platform you can simply click your system to, your chip to. You see three different chips, a heart, a gut, and a lung on the chip, and you can put them simply, instead of developing for each individual chip, developing a new design for a breadboard system, you have a standard plateau where you can just hook your device up, and you can use the same, let's say, microfluidic system that you developed once for all the other systems. And here you see how it looks like. We have here a system with 3 times 64 channels that can be connected and controlled, where the flow of fluid can be controlled, and you can, let's say, control what kind of drug you want to test. Here you connect it. Now you can just plug and play your chip in your system. We have made sure that it's an open standard so that every developer can see what is required as interconnects or as flow rates or as ceilings or as electrical connects. And in this way, you can very easily, let's say, make your chip compatible with this base system. So this is what we call the translational organ chip platform. And here you see that we have already quite a lot of partners in the Netherlands and also companies, university and companies that have contributed to these standards, and it's still under development. We also put it in the European Organ Chip Society, so it's a European, hopefully it also get the European standard. And here you see some examples of how you can control your fluidics. In the left-hand side, you see the 64 different chambers that can be connected. You can create a gradient in this case, or in the, well, it's from blue to red if you want here. Here you see you can do it in a similar way in the middle part. On the right-hand side, it will show that you can also individually fill each chamber with a drug or a liquid at wish. You see it's flowing channel by channel. And at this moment, for instance, what we are investigating is to develop chambers that are big enough to house organoids and to cultivate organoids in these microchambers. So this, we hope, is a way to combine the efforts of different groups and to benefit from each other developments. So the vision of a center that we recently started, the Organ on Chip Center Twente, is to become an international leader in the standardization of the fluidic backbone for these systems. And the mission is to educate multidisciplinary scientists to develop patient-specific organ on chips, also to standardize and validate developed technology, and also to accelerate the implementation, as you see it here. And we work together very closely with Leiden University, with 13 other universities in the Netherlands, and also different companies, amongst others, also Philips. And we have just handed in a 50 million grant proposal for, let's say, funding the infrastructure for this Organ Chip Center Twente, together with Leiden and many other partners in the Netherlands. We believe this field is really growing into a very important new tool for pharmaceutical companies, and hopefully they will soon switch over from using animals, not completely, but for a significant part, to using organs on chip. Okay, I want to just show you here the leads of this Organ on Chip Center, Anders van der Meer, Dirk van Meer, and Liliana Taxera, who are young and coming high potential people who are bringing this Organ on Chip Center to a success. Okay, the last part of my talk will be about cells and chips, and it will be motivated by the climate change as we all face it. And I always take this example as the most speaking example for me personally. The Rhone Glacier in Switzerland retreat. You see images of 1856, 1870, 1900, and 2008, and you see all the time this glacier, which was extending to the valley, down to the valley, still in 1856. Now it's uphill and you can hardly see it. It's halfway the mountain. And on a more personal note, I was walking with my daughter and my glacier a couple of years ago. We were going to the Concordia hut, which is over here, and this Concordia hut was constructed about 100 years ago, just beside the glacier, and now you have to walk up all these stairs to get to the hut. So there's more than 100 meters of ice, all melted over the years, and if you stand on this glacier, which is 5 kilometers wide, all that ice is gone. You see something is really going wrong here. While we are now suffering from the glaciers that are disappearing, in 1962, companies were still proud of their production in oil, and they compared, let's say, their production of energy, and they converted it in the amount of melted glacier that they could produce, 7 million tons of glacier. Today, nobody would dare to put an advertisement like that, but I found it kind of interesting that 6 years ago this was still on the list. So what's going on? Well, you all know this, of course, over the past few 100,000 of years, CO2 levels never exceeded 300 ppm, except for the past 100 years. And here you see it's ongoing all the time, long up, and we have to really do something to go down. Not only reuse it, which is maybe also good, but we also have to just store it. Not putting it back into the system, but just getting it out of the system. And that's what people call negative emission technologies. No emission, but just taking CO2 out of the air and immobilize it, sequestrate it, or put it into the underground or something like that. And there has been a report about negative emission technologies by the European Academy of Sciences. And here you see there's many ways you can do. You can temporarily use forest growth. You can store it underground, which is still a bit risky, direct air capture. But maybe also interesting is to use silicate rocks, like magnesium silicates, which is just taking up CO2 from the air to form magnesium carbonate. And this is then, for those interested, there's olivine sand, this is called, and there's a website called projectfesta.org who calculates that if you do 30 gigatons of this olivine rock every year, put it out, it's very slow weathering by taking up the CO2 and forming the magnesium carbonate. It's about 1 to 2 microns per year, but still it's unstoppable. It's thermodynamically going to this one way down to form the carbonate. And then maybe also in answer to David Sinton's question, what is it you use in the order of tons per year? Well, I think bricks. Bricks is used for construction material in high volumes. And there are people who are looking at the conversion of magnesium silicate powder from the high pressure with carbon dioxide to form this type of sandstone bricks. It's still energy inefficient, but it is a product that can be used and it stores CO2. But okay, this is not what I want to talk about today. I would like to talk about the ocean. You can also look at ocean fertilization. Ocean absorbs 30% of the anthropogenic CO2 in the ocean. That's also good, because otherwise it would even be higher, the rise of CO2 in the form of algae. You see here the coccolithophore, round stone bearers, if you translate it literally. These coccolithophores, they play a large role in the ocean carbon cycle. And here, left-hand side, you see also that ocean life is affected by ocean acidification. If you acidify this carbonated algae, you see that the carbon shell is disappeared. And that is, of course, something you have to avoid, because then you will dissolve even more of these calcites into the ocean. And that's why it's important to see if you can analyze the ratio between the particulate organic carbon, the POC, versus the particulate inorganic carbon, the PIC. And this PIC to POC ratio, it gives you an indication what is the ratio of the organic and inorganic, and also it has influence on the weight of the cell and also the density. So it will influence the buoyancy of these cells. So we have looked, can we develop a method to look at the peak-to-POC ratio of individual algae? And for that, we used impedance spectroscopy and in particular beta dispersion. Beta dispersion takes place at cells because at low frequency they are higher impedant than these high frequencies because the cell membrane becomes transparent at high frequency. And by looking at the ratio, you can distinguish, hopefully, between the organic and inorganic algae. As a comparison with polystyrene beads, it's a straight line. There's no difference at which frequency you measure. But with cells, of course, you see that there's a difference at which frequency you measure impedance. Now, what we have been doing is, what we've been able to do is to distinguish between calcified and decalcified cells. Here you see that the opacity, which means the ratio of the impedance at 20 and 5.5 megahertz, is different. There is still an overlap, and we're trying to exclude that. But next phase, of course, will be to online follow the production of these calcite shells of the coccolithophores. And we also found that one of the reasons why there's overlap here is that there is an effect of the height of your cell. And here you see that we can also correct for the height of the cells. And here you see in a corrected version we have a much clearer distinction between the two populations than without it. And this hopefully will work out in the future. So this is the end of my talk. I think I showed that lab-on-chip for lithium is something that is working. Organochips is a very promising concept for healthcare, and cells-on-chip can be studied to look at the carbon cycle in the ocean. And with that, I would like to end my talk.
Thank you so much, Albert. I know with your health condition, that's glad. We are so grateful that you are able to still present this really exciting talk. Let me take this opportunity to invite our next speaker, Professor Karim Karim, who is the Executive Director of the Center for Bioengineering and Biotechnology, CBB, and also he's the team lead for WIN as well. So Karim, please.
Thank you, Sushanta, for that very nice intro. Let me see if I can share my screen here. So again, I just want to acknowledge and appreciate Sushanta and the rest of the great WIN team for inviting me to give this talk, and thank you all for participating in this conference. So I'm going to talk about something that I've been working on for, I think, a little bit over a decade. And it's a technology for spectral X-ray detectors that was developed at Waterloo and is now actually, it's actually being used commercially. So that's why you'll notice that I've put the University of Waterloo affiliation, because that's where it was started, and also KA Imaging, which is the company that's actually commercialized it. So I'm going to talk to you about the world's first portable dual energy or spectral X-ray detector for the screening and monitoring of COVID-19 pneumonia. It's actually used for other pulmonary diseases, but COVID-19 is so topical, it seems to make sense. So even before COVID pneumonia burst upon the scene, lung disease was the third greatest killer worldwide, resulting in more than 5 million deaths annually. It's a problem that is as much a problem in North America and the Western world as it is in emerging nations. You can see, for example, the highest death rates due to lung disease show up in the Indian subcontinent, China, and parts of Africa. But you also have still, I would say, a non-insignificant number of deaths in North America and parts of Europe. And the reason for that, of course, is lung cancer. That's a big, big driver. But in Asia and Africa, it's primarily pneumonia that actually drives these numbers. Now, so let's talk about COVID pneumonia. COVID pneumonia, of course, came upon the scene earlier this year. And the biggest impact of this pneumonia was that people just stopped going to hospitals. So what you see here is that in mid-March, this graph actually shows how the volume of patients, especially outpatients, dropped precipitously. They're down almost 65%. And they said just in a month, the hospitals in the US were losing about $50 billion because of canceled elective procedures. And today, I think that number has exceeded $250 billion. It's quite staggering. And of course, because of this, some healthcare providers are going bankrupt. The reason, of course, that hospitals are so vulnerable to volume is because they're still operating on a fee-for-service type model. Care is episodic. If you get sick, they get paid. If you don't get sick, they don't get paid. And that's just the way the system is set up. What's interesting in all of this is, so in large part, in order to figure out if people are sick or if there's a sickness issue, imaging is used, right? Like, an imaging would include not just X-ray, but ultrasound, nuclear medicine, MR, mammography, interventional, CT, there's a whole bunch. Uniformly across the board, just like you saw the number of patients coming into hospitals decrease, you also see the amount of imaging happening in hospitals decrease. And it's gone down by, in some cases, 100%, like nuclear medicine and outpatients was just not done. But the one thing that stands out on this graph is that X-ray saw an increase in usage in the COVID days. And there's actually a really, really good reason for that. X-ray is actually the only modality that was used more, that saw an increase of 20%. Everybody else saw a decrease of anywhere from 30 to 100%. So the main reason for that is X-ray is portable. That's the main kind of takeaway. X-ray is portable, and it's a very easy to use modality. It's got a very long history associated with it. And after some months, the WHO also issued some guidance that said that just X-ray should be used with COVID patients in a wide number of scenarios. For example, your swab test gives you a negative, but the patient obviously looks sick. You better give that patient an X-ray, because X-ray has been shown to pick up COVID infection in the lungs. At least I think in 10% of the patients where the PCR test returned a negative. Or alternatively, if it just takes you some time to get a result back from a PCR test and the patient is clearly symptomatic, then you should use the X-ray to see if you can pick up the disease. It also is a key tool to determine whether to hospitalize the patient or send them home, or to take that patient and put them in the intensive care unit or leave them in a general ward. And of course, to monitor the patient over time to see if they're improving or not. The PCR tests cannot do any of that. They basically tell you you have the disease or you don't, but there's still a management and treatment aspect that is with them. So this is the reason why you see that there is an increase in the use of X-ray because of COVID. Now, just for you folks who may not be familiar with X-ray, there's actually two types. There's a fixed system, which is basically the system on the left. You have a bed, or a person can stand, and the source and the detector are kind of fixed. And then you've got the portable ones, which are actually the ones of interest for us. Because with the portable ones, you can actually take the X-ray machine to the patient. And this is what's seen a lot of use during COVID. People have basically rejigged their emergency rooms and whatnot to bring in portable X-ray machines so that the staff don't have to get too close to try and minimize the spread of infection. Now, in the X-ray system, there's two components. There's an X-ray source and an X-ray detector. Our focus has always been on the detector, and our focus has been to improve the sensitivity of the detector so that it can make a more accurate X-ray diagnosis. Just to underscore how commonly X-ray is used, 2 billion procedures annually. That tells you 1 in 3.5 people on the planet get an X-ray on average. 60% of all medical imaging is X-ray. And of that 60%, 40% of it is just X-ray. For example, things like pneumonia. X-ray technology has been around for 120 years, but it hasn't really changed. The image that was taken by Röntgen many years ago, and the image that's taken with a conventional state-of-the-art X-ray detector, there are some quality improvements, but in terms of the information that you get, there's really not a huge difference. You can still see the bones relatively clearly, perhaps more so here, and the soft tissue is obscure. You can't really see it. So we know that X-ray has had a problem differentiating soft tissue. This is a well-known issue. This is actually the main reason why other imaging modalities like MR and ultrasound and CT have gained dominance in recent years. Actually, about 40 years ago, X-ray came up with a technology called dual energy X-ray that kind of alleviated the problem of tissue differentiation, but it never really took off. It didn't become mainstream. It remained a niche technology. So what's dual energy X-ray? Dual energy X-ray is you're basically looking at the spectral information inside the X-ray beam. X-rays are polychromatic, so you have a range of X-ray energies. And with dual energy, you're actually making use of the different mass attenuation coefficients associated with various X-ray energies in the body to essentially do material separation. Now, I'll explain that in a bit. So you've got a low energy X-ray and you've got a high energy X-ray. And then these are taken at with different spectral beams. And then you do an algorithmic subtraction that allows you to get a soft tissue image and a bone image. Obviously, if you see the soft tissue image, you can diagnose soft tissue disease like pneumonia very easily because you don't have the bones hiding characteristics or adding to the confusion. And vice versa. You can look at the bones very clearly and then hide the soft tissue. And then if there's a bone disease you're interested in, you can pick that up as well. To give you a little bit more information on what we mean by dual energy and a little bit of the physics. So in this case, you've got an object sitting here, and inside this object, the soft tissue, you've got this calcification or iodinated vessel. It's just something that's of a different density. So when the X-ray photon comes in, it'll pass through the soft tissue, and it also passes through the soft tissue and the bone combination. But understandably, there's going to be a difference in attenuation because of this vessel right here, or this calcification. So you end up getting a different signal on your detector, one that'll pass through soft tissue and one that passes through this combination of soft tissue and bone. So that's an important point. Now, remember I told you that the attenuation coefficients for X-ray at different energies are different for different materials. So this graph actually gives you a nice idea of what that is. For example, at 60 keV, you have a mass attenuation coefficient for bone, which is, of course, much higher than that of soft tissue. But when I go to 120 keV, they're actually very similar. But the fact that they have this difference in the mass attenuation coefficient at 60 and 120 then allow you to use that subtraction technique to actually figure out what one material is. It's like you're solving two simultaneous equations, if you will. So the equation, the algorithm that's used is actually incredibly simple. I mean, in practice, you have to do a bunch of other things to get the image quality up. But in an ideal situation, it is actually as simple as it sounds. Your dual energy image is nothing more than the subtraction of the bone signal and the soft tissue signal with some kind of weighting factor. And the weighting factor is typically the ratio of the mass attenuation coefficients. And it's as simple as that, right? Very, very simple technology. Now, what's the benefit of dual energy? Obviously, the image quality is much better, so you're going to see early disease detection, and that's going to shorten the time to get a corrective procedure. In the case of COVID, you can be sent home to isolate, or you can be admitted. In the case of lung cancer, your treatment can start sooner. You also get better patient and operator safety. Dual energy gives about 20 times less radiation than a CT, and you have a significant reduction in diagnostic error. And in the US, it matters because it's associated directly to malpractice. You also get higher operating efficiencies. Because the images are clear, they're easy to read. So actually, even though you have 3 images, studies have shown that radiologists can read these images in 30% less time than a conventional X-ray. And then the last one is, of course, because the images are easy to read, you can utilize people with different abilities, like they may be not as experienced, but they can still get a good outcome. And of course, it's a lot cheaper than CT. So with all these advantages, one might ask, why is dual energy not being used? And there's a really very simple reason for it. And the simple reason is the technology has not enabled it to get a good workflow. So here's an example of the most popular dual energy solution in the market today. It's called KV switching. What KV switching does is it does the 2 X-ray exposures like we talked about. You get the high energy and the low energy. The problem here is twofold. One is you're taking 2 exposures. When you take 2 exposures in medical imaging, it's not like in industrial. You cannot just get a hospital to switch away from a single exposure X-ray to a dual exposure, because now you have to justify the extra radiation. So there are only limited cases where you can justify the extra radiation, and the machines are quite expensive because you have to have these special X-ray sources that can switch between low and high. So actually, it doesn't match the hospital workflow, so it stays an issue. The other problem with 2 exposures is when you take them sequentially, you can get the patient to hold their breath, but they can't stop their heart. The heart continues to beat, and you end up getting a motion artifact. And just to give you an idea of what that motion artifact looks like, here's a streak that you see in the corner right here. Problem there is you've got your 2 X-rays. It gives you the motion artifact. It's expensive. It's not portable. And it doesn't fit the workflow with its 2 X-ray shots. So you know what? The adoption is very, very low, actually. Most radiologists have only heard of dual energy, but they've never used it. Now, even before the 2 exposure solution, there was another solution in the business. It was actually commercialized by Fujifilm. And what they did is they took 2 sensor plates. They used to be CR plates, scintillators basically, and they used to put a copper mid filter in the middle to cause so the X-rays would shine on this device. Some X-rays would be absorbed in the top layer, some would be absorbed in the copper, and some would get to the bottom layer. And then you could get spectral separation by subtracting the image from the first layer from the image in the second layer. The problem there, of course, is you end up with good spectral separation, but poor dose efficiency because you lose photons in the copper. So that never took off because it wasn't an efficient system. What we did at Waterloo, which is how we changed the game, is we actually used a 3-layer approach, and instead of a copper mid filter, we replaced it with another sensor layer. So in our case, all the photons are counted, but you can still get spectral separation because you have 3 different layers, each collecting a different spectrum, hardened by the top layer. Just to give you an idea, here are the spectra for this type of device. You can kind of see how the spectra change as the beam hardens, as it goes through the different layers. But it's essentially the same thing as a dual energy device. The nice advantage, of course, of having 3 layers is you now get an X-ray detector that has the best dose efficiency in the world. Our device beats a conventional X-ray detector by about 20 to 30% just because there's more absorption of photons. And the other advantage, of course, is that you can now take this device. It's just all concentrated in one detector, and it only needs 1 X-ray exposure because the spectral separation happens in the detector. So you get no motion artifact. And this detector can be used to upgrade any X-ray machine, be it a fixed machine or a mobile machine. So basically, we changed the paradigm for dual energy. We often say we reinvented it by this approach because now we get the humble mobile X-ray that gets diagnostic accuracy that is much closer to a CT scan. That's really important, not just from a workflow perspective, but from the portability perspective. Because as you know, with COVID, if it's not portable, you can't really use it. These are just some images we took from early clinical trials in 2018 at the Grand River Hospital in Canada. We've also got another site at Princess Margaret Hospital in Toronto. They're using it for pneumonia and COVID. And here you can see the image quality. There's no motion artifact. And in this particular case, the soft tissue image was able to pick up a lesion that the bone image that the regular image was not able to. So it kind of gives you some interesting idea. And I mean, in terms of use cases, there's a whole bunch. There's lung cancer, there's pneumonia, there's COVID. There's even medical error like line in tubes called pneumothorax. So basically, our vision is that this device is going to replace X-ray in all portable settings and eventually in fixed ones as well. These are just some pictures of where this device could be used. For example, in outpatient clinics, Walmart has now gotten into the X-ray business. In long-term care facilities, this fellow here is taking his X-ray in a van to a long-term home. In vans, as well as in intensive care units and in emergency rooms. So this is where we see the future of X-ray. Dual energy can enable extreme portability, which can allow this now to see application like automatic automated external defibrillators, for example, for like cardiac disease. So we could see X-ray being used as a screening tool for pneumonia, for COVID, for fractures in places like pharmacies, nursing homes, gyms, malls. You can really see how the portability aspect has been improved for X-ray. So I'll stop here. Thank you so much, and I can take questions if there's any.
And thank you, Karim, for excellent talk, and the innovation is just fantastic, particularly in the context of COVID. So let me take this opportunity now to invite our next speaker, Professor Warren Chan, who is the Director of the Institute for Bioengineering at the University of Toronto. Warren, thanks for joining us as well.
Yeah, I'd like to start off by thanking Dr. Mitra and the committee for inviting me here to give this seminar. So I'm going to present the work that we've been doing in the last 20 years in terms of developing strategies to deliver nanoparticles in solid tumors. The other half of my lab is working on nanoparticle-based diagnostics, so I thought I'd just decide to focus on one of the two topics. As Dr. Mitra mentioned, I'm currently a professor and director of the Institute of Biomedical Engineering at the University of Toronto. So I'm going to start off with a concept that has been pervasive within the nanotechnology field for the last 30 years or so. It actually started in the late 70s, early 80s, but it really started to have a lot of attention in late 1990s and early 2000s. And that is actually the use of nanoparticles for cancer application. So this is the central concept of cancer nanomedicine, which is where the nanoparticles are designed, and the nanoparticles could be an imaging nanoparticle. Let me just get the laser pointer out. So where the nanoparticle could be designed for imaging of diseases, or for carrying drugs, or a combination of imaging and carrier of drugs. So the principle involves the designing nanoparticles and injection into patients. It was expected that these nanoparticles would circulate in the patients, and then eventually they accumulate in the tumor. So they diffuse through gaps in the tumor, and when the nanoparticle reaches the final target site, if it's a drug-carrying agent, it essentially releases drugs at the final site and then kill the tumor cells. There were essentially two advantages proposed from this particular principle design. One is that because the drug is essentially stored in a nanoparticle, it actually minimizes the side effects associated with the patient because the drug essentially do not interact with healthy tissues. The second principle is that it would actually lead to an increased therapeutic effectiveness because all the drugs would be released at that targeted site. However, despite this concept, in 30 years or so, there are very few of these cancer nanomedicines that have translated from design into patient care. So in fact, there's only about 10 nanoparticles at this particular point. So what we're at this point is to try to figure out why do nanoparticles not translate from conception into real world application. And so we're really at the phase in terms of cancer nanomedicine where we're trying to define the problems. And so this concept of evaluation of the current state of cancer medicine is actually very healthy for the field because it's been around for 30 years. And every time you develop new technology, you should always evaluate to see what the problem so you can move the technology forward. So in terms of the last 30 years, I just want to keep on reiterating this, is that what you have is that in the 1990s, you developed the concept, lots of really nice papers published in Nature, Science, and all the sort of high impact journals. And we saw very few of these nanoparticles translate into patient care. And so we're at the phase that we're defining the problem. And once we define the problems, we can find the solution. And once we have the solution, I think the clinical translation is going to be a very natural process in this field. So the biggest issue that we found a few years ago that actually we attribute to the low delivery, translation is the fact that very few of the nanoparticles you inject in are actually delivered into the tumor. So in this example here, we have 1 million nanoparticles that you engineer outside. When you inject 1 million nanoparticles in the patient, what ends up happening is that about 7,000 of those particular nanoparticles will be accumulating the tumor. The remaining nanoparticles essentially will be trapped in the liver, spleen, bone marrow, and other organs. And then if you look at the bigger picture, is that how many of those particular nanoparticles reach the tumor cells, what you see is that out of the 7,000, only 14 of them reach the tumor cells. And then if you're trying to reach a nucleus for some drugs, maybe one of those 1 million nanoparticles would essentially accumulate in the nucleus. So what my lab and a number of labs are attributing the low translation is that when you inject nanoparticles into the body, very few of those nanoparticles actually are delivered into the target site. So this presents what's called a delivery problem, right? Because you have to deliver the agent into the target site for it to work effectively. So what my lab has been doing for the last 20 years has been trying to solve this delivery problem. And the way we have been envisioning to solve this particular delivery problem is by analyzing and characterizing the nanoparticle journey. So once you inject the nanoparticles into the body, what organs are they interacting with? What molecules are they interacting with within that particular organs? What cells are they interacting with in terms of the nanoparticles? And so we're starting to look at this in a very systematic manner. So I'll give you an example here in the lab. So when you inject a nanoparticle containing drug into the system, what ends up happening is that they interact with the blood. So they circulate in the blood after IV administration. So within the blood, the nanoparticles make some decision. It can go to the liver. It can go to other MPS, myelophagocytic system organs, such as the spleen, the lymph nodes. Then it redistributes to other organs like the lung, the spleen, the bone, right? And then if the nanoparticles do not degrade, they actually retain in the body and then continue to circulate. Or the nanoparticles after blood, they can interact with the kidney, and then you can essentially pee it out. Or they can go to the liver, and then you can actually remove it through the bile system. So within your body, it actually contains, the way I view it, is a lot of individual organs that actually interacts with the particle. And when it interacts with the particle, it either alters it, metabolizes it, or chemically just modifies so that it actually behaves a certain way. But it's a very complex system. And so what my lab has been doing for the last 20 years has been looking at each individual organ and how they interact with nanoparticles, the liver, the kidney, the tumor, the blood, the lymph node, the lung. So each individual student in my lab, when they do the thesis, they actually have one focus within that particular organ system. So the idea is that once you actually understand how the particles interact with these organ systems, you can actually develop design rules that would overcome these particular organ systems. And for example, here, this is a good example of how we're thinking about this. If you design a nanoparticle, get in the blood, if you think of it from the body's perspective, it's asking this question: what is the size of the nanoparticle? If it's less than 5 nanometers, the body will essentially remove it through the kidney and you pee it out. If it's greater than 5 nanometer, it then asks the question: is the particle degradable? And it metabolizes, and the metabolic agents now goes back in circulation, and now it's smaller than 5 nanometer, it comes back out. If it's larger than 5 nanometers and it's not degradable, essentially goes through the cover cells and then goes through another route of mechanism. However, if it can't avoid the cover cells in the liver, then when it comes through the cover cells, and then your body essentially retains it. So your body essentially decides what to do with the particles based on the size in this example, but they can also make these decisions based on the shape of the particle, as well as the surface chemistry, as well as the chemical composition. So all of the physical chemical properties of nanoparticles have an impact in terms of what they do in the body and how the body processes or interacts with them. So we call these barriers. So these are barriers that actually prevent the nanoparticles from going into the target disease site, because it's essentially trying to do all these other things in addition to trying to target the tumor space. And so what my lab is trying to do is, with all these studies, is try to develop a blueprint so that we actually understand these interactions and then use the blueprint eventually to help design the nanoparticle so that it can effectively be delivered into the tumor. So what I want to share with you is 5 of the principles that we've learned in the last 15 or 20 years. And these principles are currently driving a lot of the research activities within the nanomedicine field. The first concept is the biological identity of the nanoparticle. So when you make a nanoparticle, you essentially characterize the size, the shape, the surface chemistry, the stiffness, to characterize all these kinds of physical and chemical properties of material. However, when you inject it into the body, what ends up happening is that within the blood system, for example, an IV injection, you have proteins that absorb onto the surface, which is indicated by this green blob on the surface. The nanoparticles may essentially start to aggregate in dimers, trimers, tetramers, and actually different sizes. So we call this the biological identity. And what actually happens to the nanoparticle as they circulate in the bloodstream is dependent on the biological identity and less with the exact synthetic identity. Because the biological identity is what interfaces with the cells. So in this particular example, you have the proteins that absorb on the surface. Those proteins can bind onto certain receptors on a cell. It can actually trigger a certain signaling pathway. It can trigger cell death. It can trigger toxicity. It can trigger immune response. It can trigger a lot of events. So it's actually this kind of surface that dictates it. Whether if it's a monomer, dimer, trimer, actually impacts the rate and the length of retention of the nanoparticles is or are on the surface of the cell. So when it's a one nanoparticle receptor ligand interaction, it tends to bind at usually a micro to nanomolar range. But when you start to do a dimer, there's multiple interactions, which now increase the strength of interaction, which impacts how long the particle stays on the surface or in the cell. So essentially, there's a correlation between the synthetic identity, the biological identity, and the physiological response. So we're trying to do this correlation. But the bigger picture is that this is actually really complicated. So on the right here, I just show a mass spec of these proteins that's absorbed onto the surface. So we analyze over 76 different gold nanoparticle formulations with different chemistries, sizes, and shapes. And what you see here is that there is actually no two lines that are exactly the same. So like, for example, here, this is a G15AHT and G15MHA. The AHT and the MHA refers to the surface chemistry of the nanoparticle. And what we did was we actually took gold nanoparticles, soaked it in serum with the different designs. Then we purified, we removed the gold, and then we analyzed the proteins that absorbed onto the surface. And what you see here is on the top here is all the different proteins that's absorbed onto the surface. And what you see here in this heat map is that there's no two gold nanoparticle designs with the exact same protein combination. So what that means is that when you essentially make your nanoparticles, any slight modifications you do to them will affect the types of proteins absorbed to the surface, which then affects the physiological response. And so right now, one of the big projects in the lab is trying to figure out the correlation between the physical chemical properties of nanoparticle with the protein absorption and the cell and tissue response of that particular nanoparticle formulation design. The second concept is that a lot of the nanoparticles do not target the tumor, will also be trapped inside will be trapped in the liver. So the question is, how does the liver remove nanoparticles from circulation? So in this study in 2016, we actually analyzed the mechanism of nanoparticle sequestration in the liver. And what we found is that when you inject nanoparticles in, it circulates around. And when it guts into the liver through the portal vein, the speed by which the nanoparticle is moving goes really fast. Then it gets into the little sinusoid in the liver, which is in this region. It actually slows down about 1,000 times. And when it slows down, it allows the cells in the sinusoid to be able to take up nanoparticles. So then it essentially removes the nanoparticle from circulation. And then if the nanoparticle can escape the cells in the sinusoid, it comes back out and goes back to the central vein, it goes back in circulation, and it goes fast again. So in this process, in the first round, the liver can potentially remove, let's say, 30% of the nanoparticle. Once they circulate multiple rounds, what ends up happening is that you remove another 30% from circulation. So as the nanoparticles circulate multiple rounds, what ends up happening is that fewer and fewer nanoparticles are available to target in the tumor. So one of the strategies to overcome the low delivery efficiency of nanoparticles is to essentially remove the cells in the liver sinusoid so they can't pick it up, or you can accelerate the speed by which the nanoparticles are moving so that there's more particles that can escape the cell nanoparticle interaction to get back into circulation. So the next part of this concept is that once you can escape the liver, the next aspect is how do nanoparticles transport into the tumor microenvironment. So before 2016, the mechanism is based on this leaky vessel model. So what that means is that as the nanoparticles circulate, if it does not get trapped in the liver, it essentially gets into the tumor space. And when it gets into the tumor space, it diffuses through gaps between cells, which is depicted here. When it diffuses through these gaps, it essentially now interacts cells that are buried behind the vessels. And this is actually one of the big premise of using nanotechnology for cancer applications in the 1990s and early 2000s, is that these gaps tend to be in a size range of anywhere from 100 nanometers to 400 nanometers. And nanoparticles is actually the perfect design because you can design these things small enough so they can get through the gaps. And the other aspect is that people thought that the tumor cells, at least the nano community, thought was very well organized. And we'll show later on that it's actually not that organized. It's very complicated. But the model has been sort of turned a little bit upside down in the last year or so, and I'll explain why. So what ends up happening is that the gaps is one mechanism, but the field did not look at other mechanisms of transport. So in this example here, this is the gaps. So nanoparticles, if they're small enough, they can diffuse through. The thing is that nanoparticles can also go through cells. So in this example here, a vesicle that is small enough with a gap that nanoparticles can transport through, or you have multiple vesicles that join together. And when they join together, you create a channel by which nanoparticles can transport through. So these two other mechanisms were not explored heavily in the last 30 years because everybody assumed that this is how nanoparticles get into the tumor. But because there's low clinical translation, low delivery efficiency, we and a few other groups start to explore the mechanism, because if the nanoparticles can't diffuse through the vessel against the tumor cells, that may be an explanation of why there's so little nanoparticles that have accumulated in the tumor when you inject them in. And so we published this paper a couple of months ago, which actually shows the dominant mechanism of nanoparticle transport into the tumor is not through gaps between cells, but it's actually through the cells. So this through the cells is actually an active process because the cells is always evolving. So sometimes these gaps within the cells are present, sometimes they're not. And in this particular study, and just use a TM image to depict this, is that the nanoparticles circulate in the blood vessel. And then what happens is it binds to receptors on the endothelial cells. And then it essentially goes into vesicular structures and it gets transported through the cells. And then eventually it gets dumped out at the other side, right? And the question then becomes is what happens once it gets to the other side? So that's the fourth principle I'm going to present. But the reason this is a very different concept is because in this concept, we're shown that we may potentially be able to actively design the nanoparticles so they can prefer to transport through a active mechanism versus a passive mechanism, which is diffusion through gaps. And this is other sets of ongoing studies that are happening. The next question then becomes is that what happens to nanoparticles once it gets through the vessels and into the space? And so the previous model that was shown before 2016 is that the tumor cells are nicely organized so that when you have a nanoparticle, it can diffuse through the gaps. And if you put antibodies, aptamers, or other types of recognition molecules on the surface, it can actually recognize the receptors on these particular cells, and it can accumulate in that particular point. We showed in 2018 that it's not that straightforward. And what happens when nanoparticles transport through the vessels to get into the tumor microenvironment, what ends up happening is that you actually have a lot of different cells that are located there, which is demonstrated here. The cells are not very organized, meaning that nanoparticles can interact with cell 1, cell 2, cell 3, and so on. And so then essentially it goes through, it has to get through this kind of jungle within the tumor microenvironment to get into the final cells to be able to bind, right? So it's very complicated. And in a lot of cases, I showed earlier that there's only 14 nanoparticles out of a million that bind to the tumor cells at the end, which is depicted by this teal color cell. And the reason for that is because these macrophages and other cells can actually engulf and trap the nanoparticle. So then it doesn't allow the nanoparticles to be able to move through the space to get into the final site. And I just show you an example here. This is a microfluidic system we designed to show this principle, is that you essentially have a lot of cells here. You have the vessel here. As the nanoparticles leave, what ends up happening is that it interacts with cells that are near the vessel, but the cells that are far away, there's no interaction of nanoparticles because the matrix actually blocks it from transporting deep into the tumor. The cells that's located near the vessels takes them up and prevents them from going to the one that's deeper in place. And you can see here, there's cells. We plated on the microfluidics. you have the nanoparticle, and then you see that through time, the nanoparticles are located at one region. As time goes on, it's diffused, but then you don't see any at the other end, right? And so that's why you have such low delivery efficiency to the targeted cells, because of the fact that the tumor cells typically can be located far from the vessel. So that's one reason for the low delivery. The second reason is that if you conjugate it with an antibody on the surface, you may also have serum protein. From first concept, it essentially can block that protein from also binding to the receptor. So these are some of the issues that a nanoparticle faces as it's traveling through the body. The last principle that I want to mention is that the design of the nanoparticle affects how they behave and how they distribute in the body. Just to show a very brief example here, you design two different size nanoparticles, a 15 nanometer and a 105 nanometer, and we label with a different dye. If you inject both of these nanoparticles into a mouse, you can clearly see through time they start to distribute unevenly. And the only variability we had was basically the size of the particle. So the size starts to alter how they behave inside the body, so that we view the body as basically a big sorter. It sorts the particles based on size, chemistry, and shape, stiffness, as well as other factors. And this example shows another case where you have different particle size, you have different polyethylene glycol coating chemistry on the surface, ranging from 2 kilodaltons to 10. And what you see is that the half-life is related to these designs, right? So something that's very large, 80 nanometer, with a 2 kilodalton coating has very low half-life. That means the liver will take it up very quickly. And that's the reason for that is because the serum proteins absorbs onto the surface of the particles very easily and then gets taken up by cover cells. Something that is smaller with a huge peg, there's less proteins absorbed, there's less liver uptake, and the nanoparticles becomes more available in the blood to be able to circulate, which then has a greater chance of accumulation in the tumor. So what I've done here is presented 5 different barriers to nanoparticle delivery. And so these 5 barriers are continuing to be evaluated more thoroughly. But the aspect that is many researchers are working on right now is actually developing solutions. So we know what the barriers are. We know how to design relates to the barriers. Now the question is, how do you actually develop solutions to overcome these particular problems? So that's where it comes in. And so the bigger conclusion is that there's actually a lot of exciting research in this area, and there's a lot of people working in this particular space. There's a lot of biological roadblocks to targeting nanoparticles, and we're slowly discovering the relationship between the roadblocks and nanoparticle design. And this area of research is called the nano-bio interaction. And what it does is it actually sets up the foundation for the chemical design of nanomaterials for in vivo targeting. So what that means is that if you know you have a tumor that's in stage 1, you may need a 2 nanometer nanoparticle for it to target, but a 20 nanometer will not work. But if the cancer evolves and becomes stage 5, the 2 nanometers will not work, but the 20 nanometers will work. And what we're trying to do right now is to figure out how these relationships work. And so really, the key challenge right now is actually how to actually design the nanoparticle chemistry and how to actually mediate the chemistry and the physics in the biological reactor. And we're just looking at the body as a reactor. And we're just essentially looking at these relationships. If we can control the reactor, we can control delivery, which we can control the therapeutic effectiveness. And I just have one quick slide. And so the way the future works, the way we're seeing it, at least in my lab, is that right now we're still designing the input information, nanoparticle characterization, the disease characterization, the patient characterization. And what this information will do is that it will start to build into a database system. And so once you create a database system, it essentially allows you to organize this data and look at patterns. So you can look at bed distribution patterns, pharmacokinetic patterns, delivery efficiency, as it relates to the particle design, the disease, as well as the patient. Once you build this database system, then the bigger picture now becomes this scenario where you essentially go to the hospital, you characterize the patient's disease, you input the information into the computer algorithm, which then tells the chemist and engineer to design a specific particle or to use an adjuvant for the therapeutic or delivery. Administer that into a patient, monitor the effect, and then you repeat this process. So the algorithm becomes a key principle because it allows you to guide the clinician as to what the right design is and what the optimal delivery system for this to work. And then the bigger picture of all this stuff is that this database is super important because delivery is basically the biggest limitation in using any of these emerging technologies: genome editing, synthetic genomics, immunotherapy, molecular machines, engineering proteins. And the thing is that my lab believes that nanotechnology is the future. And the reason we say that is because nano is the only field that you can actually really design nanoparticles with high precision, with very accurate size, shape, surface chemistry very easily. And we can do this at this point, but we don't actually have a blueprint to be able to guide us in terms of what's the best design for that particular disease. And that is essentially the big limitation in this particular field. With that being said, I've had a lot of wonderful students, so they're the ones who do this great work, and they're the ones who are really pushing the boundaries. I've had a lot of collaborators and very good funding support from Canada. I actually just want to mention two of your former students who were the key drivers of this research. Syed and Shrey, so they both got their undergrad degree in the nanoengineering program at Waterloo, and they have been spectacular. So they're the ones who have driven a lot of all these basic studies. Syed is now, in Jennifer Dwadna's lab at Berkeley. So it's been pretty exciting for the lab because she won the Nobel Prize, and he actually helped, participate in that process during the Nobel Prize sort of celebration last while. And then Shrey is an MD PhD student, so he went back to medical school, and then he's gonna, I think, finish up medical school in a year and start doing a postdoc and likely go into the faculty more than clinical work. So again, those two were really key to what we've been doing in Toronto. And a lot of my former students have become faculty. Stefan Wilhelm is a faculty now at Oklahoma. Leo Chow is a faculty in Toronto. So they also started some initial work about 10 years ago. And Wilson Poon is a former student who's also doing his postdoc now, but looking at the computational genomics of nanoparticle cell interaction to get deeper into the system. With that being said, thank you for your invitation. I'd be happy to answer any questions you may have.
Thank you, Warren. Such a fascinating talk, and I'm glad to see Waterloo students are also contributing to this fantastic journey that you have put forward.